Hello. Today we'll continue our lecture about the techniques, these topic techniques and applications of recombinant DNA technology. This slide summarizes all of the technique. Usually recombinant technology is used to express protein and study their structure and function, as well as study gene structure, sequence, and expression. They can be used. Biotechnology can be used to create transgenic animals and gene knockout mice. As well, it can scale up production, isolation, and purification of therapeutic proteins, DNA products. As well, it can be used in human gene therapy. The purified protein we can make from it antibodies for medical purposes. We can as well find the chromosomal location of cloned genes and detect abnormalities and copy numbers. As well, we can use it to mutate a gene and study function in the altered protein produced. And to create new genetically microorganisms and animals can be used in several applications. As well, we can use biotechnology in forensic applications such as DNA printing, and we can use it for diagnosis of human genetic disorder and infectious disease conditions. First technique is Agarose Gel Electrophoresis. I'll go fast on that because most of you are familiar with that. It's actually separate and visualize the DNA fragments based on size. Is isolated. This agarose is a sugar isolated from seaweed, and when melted in buffer solution and left to cool, it will form a semi-solid gel containing small pores like a mesh through which DNA will travel. The percentage of agarose used to make the gel it depends on what you need to separate. Usually the percentage from 0.5 to 2% max. The high percentage allow resolve smaller size of DNA fragments, while the low percentage resolve larger size. To run a gel, it is submerged in buffer solution that conducts electricity. DNA is loaded into small depressions called wells at the top of the gel. The electric current is applied through electrode at the opposite ends of the gel, and then DNA migrates according to its charge and size. The rate of migration through the gel depends actually on the size of the DNA. The migration distance is usually inversely proportional to the size of the DNA fragment. DNA migrates toward the positive pole. That's because the total charge of the DNA is negative, so that's why it migrates towards the positive pole, due to the fact that the sugar phosphate backbones make it always negative charge, so larger fragments migrate slowly and vice versa. We usually use two types of dyes, a tracking dyes and the DNA staining dyes. The tracking dyes is adding to the sample to monitor migration visually by eye, while the DNA staining dye, that we need this dye to visualize the DNA after the electrophoresis. One of the DNA staining dye is ethylium bromide. It intercalates between DNA base pair and it fluoresces under UV light. Then we can take a picture for this DNA. But keep in mind that ethylium bromide is highly carcinogenic, so the scientists now are turning to use other compounds that are less harmful. This is a represented figure showing the agarose gel electrophoresis. You can see mixture of DNA fragments of different size. They are loaded into the wells. The wells is located onto the cathode, and then once you switch on the power, the DNA fragment starts to migrate. Each DNA fragment with the size is represented by a band. You can see the band, the blue bands. The longer fragments are retarded in migration, while the shorter fragment are faster in migration. And then you can visualize band by fluorescence under UV light. You can see this is actual image of a DNA agarose gel of DNA have been cut by different restriction enzymes. And you can see we usually use a marker that is known base pair size, so to compare the size with the size of the bands with the marker. So restriction mapping can be used for sequencing. So we can digest a clone gene, the DNA fragment, with a single or double restriction enzyme. Then we separate DNA fragment via agarose gel. So you can see from the figure the difference by using the restriction enzyme, and when we use one single restriction enzyme, and then when you use double restriction enzymes. If you can see, this is the DNA which have been run on the agarose gel. So you can see there is two sites for the restriction enzyme. So once you cut by BAM alone, it will generate two sites two fragments of DNA. Once you cut by PST1 alone, it will generate two sizes as well, two sizes of DNA as well. But when you cut by both DNA fragment, it will generate three different sizes represented on the agarose gel by three bands. The second technique is chain termination DNA sequencing. Normally, we call it Sanger method. This is important, of course, to determine the sequence of nucleotide of the clone gene. These two, we use it to know the nucleotide sequence of clone gene of expression, to determine the exact structure of a gene, to identify regulatory elements such as promoter, to identify differences in gene created by gene splicing, and to identify genetic mutation. What about the reaction? The reaction consists of several components. First component is the primers annealed to the denatured DNA template, the four normal deoxyribonucleotides, and the DNA polymerase, and the special component which is called DDNTP. This DDNTP, actually, the 3' OH is replaced by 3' H, making this group is non-reactive. So once this DDNTP is being added to the polymerization is terminated. Over time, this DNTP will be incorporated in the position, and as I told you, the chain will be terminated. Then the fragments are separated by polyacrylamide gels, and then you do autoradiography because this nucleotide is radioactive label to check them, and then you read the gel from bottom to top. The sequence generated from the reaction is complementary to the sequence on the template strand in the vector, because actually the polymerization is complementary to the template. There is a quick assignment you needed to do in one page in a Word file, one page in a Word file, and just from over the internet, make this page about your opinion about the human genes or gene sequence can be patent or not. There is a controversy in this issue. For a figure, you can see this figure summarize the Sanger method. So you can see actually in a test tube, you have the first component is the template, and then the primer, and then the DNA polymerase, and then you have the normal DNTP, the four different type of DNTP, and then you have the special one is the DDNTP. You can see that it looks different. So once there is a polymerization in the bottom figure, you can see once at any time, once this DDNTP is added based complementary on the template, the chain is terminized. There is a termination of the chain, and all this fragment which is formed is labeled. You run the gel on polyacrylamide gel, and you can see these chains, this fragment which is formed of different lengths will migrate through the gel according to size. If we look closely from bottom, the sequence, you can know the sequence of the nucleotide. If you enlarge this, you can read from the bottom based on the label on the above. So the first sequence is C, then G, then A, then TT, then G, then A, okay, and so on. But keep in mind that this sequence actually is complementary to the template strand, okay? And as you can see, it starts from the 5'. Okay, so you have a laser. Another way of detecting, instead of doing the gel, you if the nucleotides have been labeled with a fluorescent dye, you have a laser and detector, and that converts the signal into computer signal, and you can see the sequence arise as a peaks. That's a computer automated DNA sequencing. The next technique is NGS, or next generation sequencing. It's the advanced way of sequencing DNA. These produce highly accurate and long stretches of DNA sequence, more than 1 gigabase of DNA per reaction, low cost, up to 200 times faster than Sanger. Roche, it's a company. Roche Roche as a company produced Roche 454 Life Systems is the first company to commercialize the NGS technology. The process actually is called solid phase method. You get the fragment genomic DNA are attached to beads loaded into multi-well plates mixed with the DNA polymerase fragment or PCR amplified in a separate water droplet in oil for each bead. It's like a multi-well plates contain more than 1 million well with 1 bead per well. Each serve as a reaction tube. So instead of doing in a million BCR tubes, small BCR tubes, you do it in a million well, in a multi-well plate per reaction. This reaction is called biosequencing. The reaction biosequencing is then used to sequence DNA on the bead in each well. In this reaction, a single labeled nucleotide, DGTP, is flowed over the well. So a single labeled nucleotide one at a time over the well. Each well contains a single bead along with primers annealing to the DNA on the bead. When a complementary nucleotide crosses a template strand adjacent to the primer, it is added to the 3' end of the primer by DNA polymerase. Incorporation of the nucleotide results in the release of pyrophosphate. This pyrophosphate, which is the PPI, initiates a series of light-releasing reaction, chemiluminescence reaction, that produce light by using enzyme luciferase. So actually, each nucleotide will generate a different light, okay? A different light based on the tag of the fluorescence it have, okay? This emitted light from the reaction is captured and recorded to determine when a single nucleotide has been incorporated into a strand. By rapidly repeating this nucleotide flow, so they flow the nucleotide one time at a time. If this nucleotide is complementary, it will bind, it will emit a signal. This signal, its color, will tell us from the detector which type of nucleotide. So you can see this figure summarize the principle. You can see that in each well there is a bead, and this bead, the DNA template attached to it, DNA on bead subjected to the biosequencing reaction. You can see on the bead they have the template, the primer anneals. You add the nucleotide with the label. It generates label. Like look down in the figure at the bottom. You can see the G generates PPI, and this PPI in the presence of luciferase enzyme will generate light, and it can be detected. And this is the NGS technology. The next technique is FISH, fluorescence in situ hybridization. In this technique, we can detect used to identify a gene of interest and in which chromosome. Chromosomes are isolated from cells, spread out on glass microscope. Use DNA or RNA probe for the gene of interest, complementary to the gene you are looking for. These probes are fluorescent tagged and incubated with a slide. This probe actually will hybridize with the complementary sequence on the chromosomes. You wash the slide and be exposed to UV light. Wherever probe has bound to the DNA, to the chromosome, it will eliminate the signal for its presence. Then you do karyotyping to spread the chromosomes over a slide, and you can see from the bottom figure that you see chromosomes with spots. That means it's a signal. So what does this fluorescence on more than one chromosome indicate? Indicate the existence of multiple copies of gene and the related sequence that may be a part of the gene. So what is the application of FISH? FISH can be used to analyze genetic disorders, can be used to determine which cells in particular organs are expressing the particular messenger RNA. The next technique is Southern blotting, and this used to determine gene copy number, gene mapping, gene mutation detection, BCR product confirmation, and DNA fingerprinting. Actually, the procedures, you digest the DNA into small fragments with restriction enzyme, separate by agarose, treat with alkaline solution to denature DNA, melting DNA. Fragments are transformed into nylon or nitrocellulose filter membrane. This is called blotting. This filter or the blot is exposed to the UV to permanently attach the DNA. The blot is incubated with the probe. It is a sequence of nucleotide that is complementary for your gene of interest. and they expose the film by autoradiography. The number of bands on film represents the gene copy number. How we can study gene expression? Techniques involving analyzing messenger RNA produced by tissue. You can use either Northern blot analysis, reverse transcription, RT-PCR, qPCR, in situ hybridization, or gene microarray. So if we continue the southern blotting, you can see here that you have a gene of interest that have an EcoR1 site in the middle and the BAM-H1 at the end. So once you do restriction enzyme, and the first one is labeled DNA marker of known size, and the second one you do restriction by BAM-H1, and the third one you do restriction with EcoR1. So actually EcoR1 is cutting inside the gene, while BAM-H1 doesn't cut inside the gene. Then you run agarose, and you transfer on a nitrocellulose membrane or nylon the blot. Then you take the blot and expose to radiography, and you can see actually the second lane shows that it is being cut by BAM-H1, but since BAM-H1 have no site inside the gene, so still the gene of interest is one single piece. But when we cut it in the third lane, when we cut it with the EcoR1, you can see the DNA is digested into two fragments. To study gene expression techniques involving analyzing messenger RNA expression, you can see here from the plot, you can see from plot 1, these are actually 3 plots of an autoradiogram from a northern blotting experiment. In the first one, you can see in which RNA from 4 rat tissues, that's a 4 rat tissues, this RNA from the seminal vesicle, 2 from the kidney, 3 and 4 from the epididymis. Okay. So you can see when you run agarose gel from RT-PCR experiment, in which RNA from rat tissue was reverse transcribed and amplified with primer of actin for actin defensing. Northern blotting. The basic method is similar to southern blotting, and they can compare the quantity amount of messenger RNA present in different samples. The procedure actually isolate the RNA from your tissue, separate by gel electrophoresis, blot on the membrane, same membrane, hybridize by probe, same principle, expose band on autoradiography, and show presence of messenger RNA for gene of interest as well. So actually northern blot is for mainly for RNA, while the southern blot for DNA. RT-PCR actually is used to study messenger RNA level when level of detection is below that of northern blotting. The procedure actually pretty the same, except you're doing PCR, isolate messenger RNA, do reverse transcription to make double-stranded cDNA, use PCR with a specific primer to amplify the gene of interest, run Agarose gel to separate amplified fragments, and then you determine the expression pattern in the tissue. The amount of cDNA produced on RT-PCR reaction of a gene of interest reflects the amount of messenger RNA and the level of gene expression. Thank you.